Hello everyone, today we are going to study about CRP. What is the full form of CRP is that it is a capsular polysaccharide reactive protein. What is CRP? CRP is a pentraxin protein. Why it is called pentraxin protein? Because it is made up of 5 subcyclic units and it is a acute phase reactant protein. Why it is synthesized and how it is synthesized? It is synthesized by hepatocytes. When does it synthesized? It is synthesized mainly in the bacterial infection. When bacterial infection occurs, then there is recruitment of neutrophils, macrophage, right? In this condition, C-reactive protein is increased. In fungal, in parasitic reaction, in viral reaction, it is not increased or it is normal. If increased, it may be slightly, but in this reaction, it is normal. Because why in viral infection, CRP is not increased? Because in the viral infection, there is intracellular toxin. This intracellular toxin increases interferon gamma, interferon alpha, and in this condition they activate cell mediated immunity. But main thing to increase CRP is interleukins, mainly interleukin 6, but it may be supported by when there is rise in interleukin 1, tumor necrosis factor, means that TNF alpha. These are the things that need to be raised to increase CRP, but in these infections there is not increased in these interleukins, so CRP is not increased. How these interleukins increases the CRP? Let's see this. In this bacterial infection, there is activation of macrophage, T cells, neutrophils. So this increases interleukin 6, interleukin 1, tumor necrosis factor. This main is interleukin 6. This interleukin 6 activate Janus kinase or this signal transducer activating transcription 3. This transcription pathway is activated by this interleukin 6 in the hepatocyte cells and enters the nucleus of hepatocytes, where it activates the hepatocytes to produce CRP. I mean to say that when JAK-STAT3 pathway is activated, then this activates this CRP gene. This activates CRP gene in these hepatocyte cells and activates hepatocyte to produce CRP. Okay, hope you have understood it. And this CRP bind to phosphocholine in the bacterial wall, in the apoptotic cells, damaged cells, and when it bind to this phosphocholine, then there is the activation of complement system, mainly the classical complement system, and this helps in phagocytosis of this bacteria. It also acts as opsonin. Okay, this is the function of the CRP and this is the mechanism how it is increased. I have told you, let me repeat it again. When there is bacterial infection, there is the activation of macrophage, T cells, neutrophils. This activated macrophage produce interleukin 6, interleukin 1, TNF alpha. This activated mainly the interleukin 6 activate JAK-STAT pathway that that enters the nucleus and activate CRP gene in hepatocytes. That CRP gene produce CRP. That CRP binds to phosphocholine in bacterial wall and apoptotic cells, damaged cells. That helps in activation of classical complement system and helps in phagocytosis. Also acts as a opsonin, means that it attracts the macrophage and helps in the phagocytosis of that bacteria. So this is the function of CRP. And another is duration. In 6 to 8 hours it is increased, but it is peaked at 24 to 48 hours. This is about the CRP. I hope all of you have understand about the CRP and why it is increased in the infective state. Thanks for watching.